All right. So name the hormone secreted by the beta cells in the islets of Langerhans of the pancreas and give its function. So the responses were split between C and D. The correct answer is C, insulin. So remember, the beta cells secrete insulin, the alpha cells secrete glucagon. Insulin is the hormone of the fed state, the absorptive state. So it's responsible for decreasing blood sugar levels. Glucagon is the hormone of the fasted state or the post-absorptive state. And so that is going to be responsible for increasing blood sugar levels from storage. So the answer here is C. All right, 10 seconds left. Okay, so the responses here, again, split somewhat between B and D this time. Which of the following statements about the post-absorptive state is not correct? So let's go through these one by one. It is primary catabolic. Catabolic means to break down, right? Anabolic means to build up. So the post-absorptive state is indeed catabolic. It's breaking things down to maintain blood sugar levels in a period where there's no food. B, its primary function is to maintain plasma glucose levels, also a true statement. C, there is cessation of nutrient absorption, also a true statement, right? It's more responsible for breaking down those stored nutrients as opposed to immediately absorbing the body's needs. So that is also true. So the correct answer here is D. Insulin is actively secreted, right? We just described this in the previous question, so that is not true. Glucagon is the hormone of the post-absorptive state. So glucagon is actively secreted in this period. Insulin is going to be secreted in the absorptive state instead. All right, so let's go ahead and shift gears here. Let's move to the muscle physiology. I'm going to open up the new packet here. This is part 1 of muscle phys. And we're going to look at, again, the first 2 objectives here before the exam. So this is typically how far I usually get before we get to the exam. So basically excitation, contraction, coupling, the cross bridge cycle, and then the basic structure of muscle is what we're going to aim to complete. Okay, so let's just kind of start with a big picture. And let's talk about the anatomy of skeletal muscle first. But first, let's discuss muscle. So what are the 3 types of muscle? Who can tell me in the chat? What are the 3 main types of muscle in the body? Excellent. So skeletal, cardiac, and smooth, right? Of those 3 types, we know that skeletal is the only one that we can control. Cardiac and smooth are involuntary, so those are controlled automatically by the autonomic nervous system. So what are some of the functions that skeletal muscles help us do? What are some of the functions of daily activity or daily living? Awesome. So support and move our bones. That's a big one. Absolutely. Any others? And this is not a trick question. Okay, so yes, movement is one of the main functions of skeletal muscle. Anything that we can voluntarily move on our body, on our anatomy, is controlled by skeletal muscle. So blinking your eyes, vision is partially facilitated by the ocular muscles, which are skeletal muscles. Movement of fingers, toes, jumping, running, speaking, right? The laryngeal muscles, the oral muscles, all of that, all of our major body functions that we can voluntarily control are controlled by skeletal muscle. And in our previous unit, we talked about how that muscle was innervated. The somatic division of the peripheral nervous system, the motor neurons, which were going directly to muscle, right? So there's no separate neurons as we see in the autonomic nervous system. There is one continuous neuron, motor neuron, that innervates or excites skeletal muscle. So that's kind of the big picture of things we've talked about before. So let's zoom in here and talk about the function and more so the structure of what skeletal muscle looks like. And then we'll talk about how that innervation actually takes place. How do we go from having an action potential on a neuron to actual contraction of muscle? So the structure of muscle is that it is connected by two or more bones with the help of a tendon. So all muscles are usually attached to a non-contractile portion, which is called the tendon. And if we look at muscles, they're typically suspended between two bones. So if we look at the biceps, for example, it's one of my favorite muscles, so I'm gonna use that as a reference point. It is attached to the scapula proximally, and then all the way down onto the humerus distally. So the biceps muscle is one of the flexors of the arm. So it's going to be suspended across those 2 points in order to be able to shorten and lengthen and actually bring about its function. If we look more closely at a single muscle, what I will refer to as a muscle belly, it's going to be divided into some further subdivisions based upon connective tissue that's covering each of those subdivisions. So the epimysium is the connective tissue that surrounds the entire muscle and the tendon. Right? Notice the tendinous portion up here. Typically, there's a proximal tendon. There's also a distal tendon as well. And that is entirely covered by this epimysium. And epi means above, so that can be a prefix to help you remember exactly where this layer is. The perimysium extends deeper into the muscle belly or muscle body, and then it further divides that entire muscle into groups that we call fascicles. So a muscle fascicle is a subdivision that can be found within the muscle belly itself, and that is surrounded by the perimysium, a slightly thicker layer of connective tissue. Now, if we look inside a muscle fascicle, which is just this little bundle here, the units found in a muscle fascicle, about 100 to 100,000 of them, is the endomysium covering a single muscle fiber. So a single muscle fiber is the smallest unit of a muscle. It is synonymous with the cell, a muscle cell, and that is covered by the endomysium. Again, the prefix endo here meaning inside or the inner parts of the structure. So the endomysium covers a single muscle fiber or muscle cell. Several muscle fibers make a fascicle. The perimysium covers the muscle fascicle or muscle bundle, and then the epimysium covers the entire muscle. Now notice, and I want to point this out early, this is not the end of the structural divisions of the muscle. So although we talked about the muscle fiber as being the functional unit of muscle, the actual muscle cell, the cellular level, we have further subdivisions within a muscle cell, which are the myofibrils and then the sarcomeres. And so we'll talk about those going forward, but I want you to think about the muscle fiber as being the cellular level of muscle organization. Let's talk about some of the terminology here with muscle and some of the unique features of skeletal muscle. It is multinucleated. We will look at cardiac and smooth later on. Those are uninucleated, so they have a single nucleus. Muscle, on the other hand, has several different nuclei. So if you go back to this little schematic, we can see that although this is one cell, there are several different nuclei that are pushed off to the edge or the periphery of the cell that we can see. The sarcolemma in muscle is the unique term for the plasma membrane. So it's very similar to what we call the outer border of the cell unit. The sarcoplasm is synonymous with the cytoplasm, which is that fluid-like aqueous environment inside the muscle cell itself. So these are just unique terms that are given to muscle cells, but they are similar structures for any other cell in the body. Let's look at the cellular level and some of the structures that we can see. So within a muscle fiber, again, down to the level of a cell here, we have many myofibrils packed inside of this muscle fiber. So here's what a myofibril looks like. Other things that are packed into the cell is the sarcoplasmic reticulum, okay, which is this sort of blue structure that's spanning all around each of the myofibrils. The sarcoplasmic reticulum is synonymous with the smooth endoplasmic reticulum in any other cell. This is also going to be the storage area for calcium, and calcium is going to be very important for the regulation of muscle contractions. Also within the muscle cell or the muscle fiber are mitochondria. Now, what do you think is the importance of mitochondria in muscle? What is the mitochondria doing here inside the muscle fiber? What's the function? And why would we have a lot of it? Why would we need to have a lot of mitochondria in muscle cells? Feel free to take a guess. Doesn't matter if you're wrong. For energy, excellent. Yeah, so you're spot on, correct. Energy is super important for skeletal muscle. Again, this is voluntary movement. So muscle needs to be very responsive to our intentions, right? So when we think about moving something, that needs to happen right away. So there needs to be lots of ready supply of ATP that is made in the mitochondria for muscle contraction. So there's a lot of mitochondria interspersed throughout the muscle cell as well. Other things that we see are the T-tubules, the transverse tubules, and these are these green sort of pipelines or tunnels that are running throughout the muscle. We'll talk about their function coming up later on. This is what a cross-section view of the T-tubule looks like. So it actually goes deep into the muscle cell itself as well, in through the myofibrils. And this is really for propagating the action potential all throughout the muscle very quickly from the plate of where that muscle excitation is received. So from the neuromuscular junction, the action potential needs to go not just on the surface, but deep throughout the muscle into the myofibrils very quickly. And so the T-tubules help to propagate that action potential. The lateral sacs are the terminal cisternae. So these are the areas where the sarcoplasmic reticulum, so remember this blue structure is the sarcoplasmic reticulum, but these are the areas of that structure that come adjacent to the T-tubules. So on either side, there's a lateral sac, and together, all three of these structures, the lateral sacs of the sarcoplasmic reticulum and the T-tubule inside, this creates a structure called a triad. Okay, and we'll talk more about this function of this structure, but again, it's going to help the action potential get from the neuromuscular junction throughout the muscle via the T-tubule, and then because it's adjacent to the lateral sacs, it's going to stimulate the flooding of calcium from its storage in these sacs or cisternae out into the muscle sarcoplasm, where it can really be involved in muscle contraction. Looking more closely at the myofibrils are what give skeletal muscle and cardiac muscle their striated appearance. So this is why muscle has that elongated sort of stripped or striated appearance to it. And so it's a way for us to organize the filaments that are actually responsible for the contractions. So there's thick filaments and thin filaments. They are called actin and myosin. Actin is the thin filament. Myosin is the thick filament. And so the way that these filaments are arranged or organized is what actually creates the myofibril. And like I said earlier, gives that striated appearance to the muscle. This is why we refer to cardiac muscle and skeletal muscle, as striated muscle because of their appearance, because of the sarcomere. These striations are basically the overlapping of these filaments along the length of the myofibril. And so if we think about the actual unit of contraction in muscle, it's going to be the sarcomere, and the sarcomere are repeating along a myofibril. So here's what one myofibril looks like if you were to pull one of these units out of the muscle cell. This is a myofibril. And then if we look more closely at what's happening on the myofibril, it's repeating units of what's called a sarcomere. So this individual unit is one sarcomere. Here is another sarcomere. Here is another sarcomere. So we have several sarcomeres repeated along the length of a myofibril. And then as we mentioned before, what creates the sarcomere is the overlapping of the thin and thick filaments. Okay, so looking more closely at that single sarcomere, right? So let's kind of zoom out here for a little bit and kind of remind ourselves of what level we're at. So we have our entire muscle. We peel away the epimysium. We've got several muscle fascicles. We take one of those muscle fascicles, which is covered by the paramysium, and we pull out a single muscle fiber, right? We take one single muscle fiber, which is covered by the endomysium, and we take out a single muscle myofibril, right? So the myofibril is now at the subcellular level. So we're looking at a single myofibril here. These repeating units on the myofibril are what we refer to as the sarcomere. And the contractile proteins, which are actin and myosin, are sort of staggered or scattered, overlapping along the length of the sarcomere. Now, the way that we can describe the sarcomere is by bands and zones. And I'll talk about how we came up with this system, and then we'll actually talk about the individual bands and zones. So before, when early physiologists and histologists were looking at muscle, they were able to identify different staining patterns in the way that muscle appears. So if you look at the histology staining down here, notice that we have some really dark areas, right? Like this on the sides here. We have some really light areas, like what we can see in between here and here as well. And then we have some somewhat darker gray areas, right? So these are somewhat dark, not as dark as these peripheral areas, but not as light as these in-between areas as well. So these patterns of light and dark staining was the first way that we were able to appreciate and observe the sarcomere and the different units that are found on the sarcomere. So the area where we have overlap of thin and thick filaments is called the A band. So this is that moderate gray area that's somewhat dark, but not as dark as the Z lines here. This is the area where the actin filament actually overlaps with myosin. And so it's going to stain a little bit darker than actin alone, because actin is a thinner protein, but it's not going to stain as dark as the Z discs where we have those proteins adjoined. The H zone is considered the area in the middle where we don't have overlap, right? Notice it stains a little bit lighter, excepting for the very center, which is the M line. And the M line is where we have the myosin filaments attached along the center. So in the H zone, there is no overlap. That's why it stains slightly lighter. The M line, as we mentioned before, is the line down the middle, which is only thick filaments that are attached along the center of the sarcomere. The I band is along the periphery of the sarcomere. So this entire region here is called the I-band. It is made of thin filaments only. This is why it stains so much lighter than the H zone or even the A band, because it is only thin filaments. Actin is a thinner protein, and so it's going to stain much lighter or less dense on histology staining than myosin or any areas where there's overlap. The Z lines are linking the thin filaments. So it's an actual area where the thin filaments are joined along the center of the I band, and that is referred to as the Z line. It's going to be the most dense area because it's actual attachments of the I bands, or of the actin filaments, excuse me. And so the sarcomere is going to be the functional unit in between one Z line and another Z line. Right? So in between this Z line and this Z line, this entire region is referred to as a single sarcomere. If we look at the actual schematic here, here's the Z line, and notice how it has that zigzag configuration because the actin filaments are sticking out in both directions. In other words, the actin filaments here are helping to form the I band of this sarcomere as well as the I band of this sarcomere. So it's helping to form I bands of both adjacent sarcomeres. Notice in the center we have the M line, which is the area of attachment for the thick filaments, which are in orange here, right? Again, a much thicker protein along the center. And then the arms of the myosin or thick filaments stick out and then overlap with the arms of the actin or thin filaments. So this entire area here on this side and on this side is the A band. It's the area where myosin, which is thick, and actin, which is thin, both those filaments actually overlap. All right. If we were to take a cross section of a myofibril, we could easily observe the thickness or the difference in thickness between each of these filaments. So again, notice how the thick filaments are shown in orange. This is myosin. And then the thick filaments shown in blue are the areas where we have actin. And so this is also helping to explain the different staining patterns that we see because of the different densities or thickness of these filaments.